This presentation is based on the research article entitled Neurovascular Coupling Under Chronic Stress is Modified by Altered GABAergic Interneuron Activity. This research is important because chronic stress is a known factor in the progression of major depressive disorder, also known as MDD. Understanding how chronic stress affects the brain and neurovascular coupling function in mice can help to understand the progression of major depressive disorder in humans, and this work can help those that suffer from depression. Previous research indicates that chronically stressed rats exhibit a decrease in cerebral blood volume response to a sensory stimulus. One of the chronic stress-associated neuronal alterations that is widely studied is GABAergic interneuron dysfunction. GABAergic interneurons were greatly reduced after exposure to chronic stress, and this information can be useful for our current study. Neurovascular coupling, also known as NVC, is the tightly linked relationship between local neural activity and subsequent changes in cerebral blood flow. It ensures normal brain function by maintaining brain homeostasis and enables the microvascular system to meet the metabolic demands created by neural activity. Excitatory neurons, inhibitory neurons, glia, and cerebral vasculature are all components of the NVC unit. NVC is also important because the interplay within these units regulate adequate blood and nutrient supply to localized brain regions. Chronic stress conditions were created by placing 8-week-old male mice in plastic bags for 6 hours a day. Within those 6 hours, they were not allowed access to food or water. This process continued every day for 3 weeks. On the other hand, the control group had normal access to food and water and spent no time within the plastic bags. The elevated plus maze test. Representative heat maps showing the exploration of mice during a 5-minute EPM test are shown. Each image represents data from a single mouse, with the control mouse being shown on the left and a stressed mouse being shown on the right. Red represents more time spent in an area, while blue represents less time spent in an area. There is one open arm of the maze and one closed arm in the maze, which is shown in the control image. The image on the left shows the data of the average duration of time spent in each area of the EPM, while the image on the right shows the comparison of baseline plasma corticosteroid concentrations data. As seen in the data, stressed mice showed more fear than the controls, as they were less willing to explore the open arms of the maze. Baseline corticosteroid concentrations were elevated in the stressed group, indicating that hypothalamus pituitary adrenal axis regulation was disrupted in those mice. Shown in this slide are the experimental settings for in vivo optical intrinsic signal imaging. Light reflected from the exposed forelimb region of the primary somatosensory cortex was filtered at the 546 nanometer wavelength, which is a measure of the hemoglobin known as HBT. To assess the NVC relationship during functional hyperemia, local field potentials, or LFPs, were measured simultaneously with the OIS imaging. In vivo OIS imaging experiment data. Figure B shows the representative HBT weighted OIS images during forelimb stimulation in the control on the left and stressed mice on the right. Each image represents the average of 18 trials from a single individual. Overall, chronically stressed mice showed significantly reduced stimulus-evoked changes in HBT. As seen in Figure C, peak HBT response was significantly lower in the chronically stressed mice. In addition to Figure C from the previous slide, Figure D also shows that the average of the maximum evoked HBT response was lower in the stressed group of mice. Figure E represents the comparison of the area of activated region for the time point at which the maximum HBT change was observed, which shows that the spatial extent of the stimulus evoked HBT response was smaller in the stressed animals. Additionally, the onset of the HBT response tended to be delayed in the stressed group as seen in Figure F, and the time to half maximum of the peak was slightly less in the stressed animals, seen in figure G. Figures H and I show that there was no stimulation-induced change in heart rate and no significant difference between the two groups, being controlled and stressed. In addition, figure J shows that there was no difference in mean blood pressure, which means that it is evident that chronic stress does not alter cardiovascular effects under these experimental conditions. Altered GABA-A receptor-mediated vascular responses. GABA is a major inhibitory neurotransmitter that is modulated via GABA-A and GABA-B receptors. GABA-A is a ligand-gated ion receptor with a chloride ion selective pore, whereas GABA-B is a G protein-coupled receptor. Each GABA receptor type has been reported to affect NVC and is therefore crucial for our study. Artificial cerebrospinal fluid, or ACSF, circulation is shown in black for the control group or red for the stressed group, and treatment of a drug is shown in blue. In control mice, selective GABA-A receptor blockade via bicacolin induced vasoconstriction, which is shown in figures A and E, while selective GABA-B receptor blockade with the CGP 55845, a GABA-B antagonist, produced no significantly different responses in vascular dynamics, which is shown in figures C and G. Ultimately, these observations corroborate the prominence of the role of GABA-A receptors in maintaining normal NVC. Alteration of neuronal nitric oxide synthase mediated vascular responses. Different subtypes of GABAergic interneurons control vasodilation by releasing nitric oxide and vasoactive intestinal peptide. To evaluate which GABAergic interneurons create dysfunctional hemodynamics in chronically stressed animals, NVC under specific GABAergic interneuron subtype agonists and antagonists was investigated. Somatostatin in BIBP 3226 had little effect on vascular response compared with the ACSF condition in either the control group in figures A and B or the stressed group in figures E and F. In contrast, figure C shows that vasoconstriction was strongly induced in the control group with L-NPA, which is a selective N-NOS receptor blocker. Unlike the control group, the stressed mice showed similar vascular response, even in the presence of L-NPA, as seen in figure G. N-NOS is one of the isomers of the enzyme involved in the synthesis of nitric oxide, which is a major vasodilator that regulates CBF. Therefore, the N-NOS-mediated vasodilation pathway is likely to be impaired by chronic stress, both in mice and humans. Chronic stress was associated with both attenuated hemodynamic response and LFP response to forelimb stimulus. Collectively, results suggest that GABA-A receptor-mediated vascular response is affected by chronic stress in mice. This study demonstrates that NVC is disrupted during functional hyperemia in chronically stressed animals. Additionally, GABA receptor agonists induced significant differences between the control and chronically stressed groups, which indicated a strong correlation between chronic stressors and GABA-mediated vascular response. Chronically stressed mice exhibit a reduction in N-NOS expressing neurons. Findings strongly support that NOS interneurons have an important role in maintaining NVC. Critique and future studies. Overall, this was a well-done study to fully examine the proposed hypothesis. It included very interesting data that can be beneficial for further understanding of the stress within college students and professors alike. However, the information cannot directly be applied to humans, and it must be translated from the mice. As for future studies, expanded research can include humans using these known results from the mice. However, the causes and levels of chronic stress will be different from person to person, so this must be taken into account for correct data. Citations for images and the research article used.